Welcome back to this next video, and in this video we are going to talk about the drug designing or the drug discovery. So the first thing is, what is drug design or what is drug discovery? Now, drug design, also known as the rational drug design or simply the rational design, that is actually the inventive process of finding new medications based on the knowledge of a biological target. This is how you are going to define the drug discovery or the drug design. Now, in most of the cases, the drug is most commonly a small organic molecule that either activates or inhibits the function of a biomolecule. For example, if that biomolecule is a protein, so this small organic molecule may be activating or inhibiting the function of that particular protein, and the activation or inhibition of this particular protein, this in turn will result in a therapeutic benefit to the patient. Now, in the most basic sense, this drug design, it involves the design of molecules that are complementary in shape and charge to the biomolecular target with which they are going to interact and therefore will bind to it. Now, drug design frequently, but not necessarily, relies on computer modeling techniques, and when you are going to use these computer modeling techniques, the drug design can also be referred as to the computer-aided drug design. Now, in addition to the small organic molecules, the biopharmaceuticals, especially the peptides and the therapeutic antibodies, they are an increasingly important class of drugs. And the computational methods, these computer-aided methods, that actually helps in improving the affinity, the selectivity, and stability of these protein-based biotherapeutics. Like these computer-aided methods, it can help you in the improvement of the drug-like properties of the peptides and the antibodies. Now, this phrase drug design is to some extent a misnomer, because a more accurate term that will be a ligand design. Because when you talk about the interaction of a protein with its ligand or with its target, we actually use the term ligand. So the ligand design, that is a more appropriate term as compared to the drug design. Before moving further, the question is why we need to develop new drugs. This is the most basic questions, because if you are developing new drugs, there should be some kind of justification to it. So why we need to develop new drugs? Now, one of the things that compels you for the new drug development is the emergence of a new disease. If there is a new disease, that means you have to go for a new drugs so that you can actually tackle that particular new disease. It is one of the most basic thing that compels you to go for the new drug development. The second important thing is the antibiotic resistance, and this antibiotic resistance phenomena, it is one of the most you can say the most complicated problems faced by the medical science in the current era. And what this antibiotic resistance mean is that the antibiotics that you once used to control the disease caused by a pathogen, now these antibiotics are no more able to control those particular pathogens, because the pathogens, they have modified themselves. So if they have modified themselves, that means you have to go for the new drugs. So the antibiotic resistance, that is another reason why you need to go for the development of the new drugs. Now, another important reason is that there are advancement in technologies that can actually give you new drugs with fewer or theoretically no side effect. That is not now, this is no, forget about this W. So advancement in technologies that can actually give you drugs with fewer or theoretically no side effects, because when you talk about the available drugs, they may be curing the disease that are used for, but they can also be causing some side effects. So advancement in the new technologies, if they can give you new drugs with fewer or theoretically no side effects, that can be very good drugs. Now, this design of the techniques, although this design techniques for prediction of binding affinity that are reasonably successful when you are going for the, say for example, if you have got a protein and if you want to use a ligand, that whether that particular ligand is going to interact with that particular protein, and if the ligand is able to interact with that particular protein, you may predict that that particular ligand can be used as a drug for that particular protein. But beside this, there are many other properties that should be taken into consideration before declaring a ligand that can actually become an efficient or the efficacious drug. These important properties, the first important property is known as the bioavailability. Now, what this bioavailability mean is that the fraction of an administered drug that reaches the systemic circulation. Say, for example, if you are taking a drug, how much of the fraction of that particular drug is actually available for interaction with its target. So this is known as the bioavailability. So this is one of the important property that you should keep in mind when you talk about the drug and the target interaction. Another important property is known as the metabolic half-life. This metabolic half-life is also known as the biological half-life, the elimination half-life, the pharmacologic half-life of a biological substance. So this metabolic half-life actually means that the the time it takes from its maximum concentration to half maximum concentration in the human body. So this is known as the metabolic half-life, and this is one of the important thing when you talk about the drugs. You should also have to take into consideration the side effects. Say, for example, if a drug is if a drug is good at inhibiting its target or activating its target, but if at the same time it is causing serious side effect that can actually go on the negative side of that particular ligand or that particular drug. So the side effect should also be taken into consideration. Another important property is known as the affinity and selectivity. What I mean by affinity and selectivity is to reduce the potential of a side effect. Another important property is the efficacy or potency, which is actually the measure of drug activity expressed in terms of the amount required to produce an effect of a given intensity. So this is not only about the interaction of the ligand and the target, but you should also have to take into consideration all of these properties. All of these properties should be optimized before a ligand can become a safe and efficacious drug. Now, these characteristics, they are often difficult to predict with the rational drug design techniques, and you have to go for the animal models and the clinical trials that we will discuss in details. Now, nevertheless, due to the high attrition rates, especially during the clinical phases of the drug development, more attention is actually given to the early phases in the drug designing on selecting candidate drugs whose physicochemical properties are predicted to result in fewer complications during the drug development and hence more likely to lead to an approved marketing drug. I'll tell you in the next video that when you start the development of the drug, you usually start on a very small scale, and on the very small scale you have to optimize all of these conditions before going into a higher scale, because economically or in terms of cost, these small level experiments, they are cost effective as compared to the large scale experiment if you are going to experiments on the animals or if you are going for the clinical trials. Now, furthermore, in vitro experiments, as I've told you, the in vitro experiments complemented with the computation methods that are increasingly used in early drug discovery to select compounds with more favorable ADME and the toxicological profiles. Now, what this ADME mean? That this A is for the absorption, this D is for the distribution, this M is for the metabolism, and this E is for the secretion. So these are the important things that you should keep in mind when you talk about the properties of a drug. That how much of the drug that is actually absorbed, how this particular drug is distributed in the body, how it is metabolized in the body, and hence how it is excreted from the body. And the toxicological profiles, you can simply say the side effect profiles of these drugs. Now, the sequencing of the human genome, it has allowed for a rapid cloning and synthesis of large quantities of purified proteins. Now, if you are having a large quantities of the purified proteins, that means you have to go for the high throughput screening. And this high throughput screening method, it is one of the most common practice when you talk about the identifying of the ligands or identifying of the hits for a particular target. Now, this high throughput screening means that if you are conducting millions of chemicals, genetic or pharmacological tests in a single go. So what this high throughput screening do is that in this high throughput screening, you will be screening millions of the compound libraries against the isolated biological targets. We'll be discussing about the different types of the targets in a while. So these high throughput screening, it actually allows you to screen millions of compound libraries against the isolated biological targets, and these biological targets that have been hypothesized to be disease modifying, or these are the targets they are actually responsible for causing a disease in the human beings or in the animals. Now, when you go for this high throughput screening, what you do is that you are going for the identification of the hits, and the hits from these screens are then tested in cells and then in animals for efficacy. Now, why the identification of these hits is important? Because if you are screening, say for example, million of compounds, maybe thousands of compounds, they are actually interacting with the biological targets, and the those particular ligands out of these millions, only these thousands or you can say 100 of these ligands that will be then screened in the cells or in the animals. So this high throughput screening is actually you can say giving you the ligands which are having a very favorable kind of interaction with the biological targets. Now, when you talk about this modern drug discovery, it is actually a very capital intensive process that involves large investment by the pharmaceutical industry as well as the national governments who provide. These national governments, they actually provide the grant and the loans. Now, despite the advances in technology and understanding of the biological system, this drug discovery is still very lengthy, expensive. Like it is going to cost you in billion of US dollars if you are going to market a drug. This drug designing is very difficult and inefficient process still in 2021 with a low rate of new therapeutic discovery. Now, this drug discovery is done by the pharmaceutical companies with the research assistance from universities, and most of the research on this ligand target interaction or this ligand target interaction is actually carried on by the research associate and the professors in the universities. And the final product of the drug discovery is actually a patent on the potential drug. Now, this drug, the drug requires very expensive phase 1, 2, and 3 clinical trials which are conducted in human beings, and most of these clinical trials, they fail. Thereby, we say that the modern drug discovery is a very expensive process. Now, small companies that have got a very critical role often then selling the rights to larger companies that have the resources to run the clinical trials. So what these small companies do, that they can actually take the drug and they can actually do experiments in the animals, but they do not have the funds to conduct the experiments in the human beings. So what they do is that the small companies then they sell the rights to the larger companies who have got a very large budget of the R&D. Now, the drug discovery, discovering the drugs that may be a commercial success in most of the cases or a public health success, but this health success or this public success that involves a very complex interaction between the investors, the industry, academia, the patent laws, the regulatory bodies, the marketing, and the need to balance secrecy with communication. So as you can see, a lot of the persons from different background that are actually involved in the discovery of a drug. Now, meanwhile, there are some kind of disorders which are very rare, and the drugs that are actually used to treat these rare diseases, they are known as the orphan drugs. Now, why they are called the orphan drugs? Because when you talk about the prevalence of these diseases, they may be present in like one in a 0.5 million or 1 in a million. So the companies, they actually do not invest in these kind of the drugs, but the drugs that are developed for these rare disorders, they are known as the orphan drugs. And these orphan drugs, they are used to treat the orphan diseases. If I give you an example of this, one of the drug that is known as the arginate, which is used to treat the acute intermittent porphyria, and this particular AIP disease is actually the deficiency of the enzyme known as the hydroxymethylbilane synthase, and the symptoms of this particular disease that includes the fast heart rate, the muscle weakness, there is pain in the arms, legs, chest, neck, or heads. So this is an example of the orphan disease, and this is the example of an orphan drug. Now, the orphan drug funding process ensures that people who experience these rare disorders, they may have some hope of pharmacotherapeutic advances. Now, when you talk about the drug targets, the definition of the target itself is something argued within the pharmaceutical industry. Now, generally, the target is the naturally existing cellular or molecular structure. It can be a protein, it can be the DNA, it can be the RNA. So, the targets, they are actually the naturally existing cellular or molecular structures, and these structures are involved in the pathology of interest, in the pathology of that particular disease that the drug in development is meant to act on. However, the distinction between a new and established target is very important. Now, this distinction is typically made by the pharmaceutical companies engaged in discovery and development of the therapeutics. Now, what the new target and what the established targets means, we will come to that in a while. Now, in an estimate from 2011, 435 human genome products, they have been identified as therapeutic drug targets of FDA-approved drugs. Now, when you talk about the established targets, those established targets, they are those for which there is a good scientific understanding supported by lengthy publication history of both how the target function in normal physiology and how that is involved in human pathology. So you have a very good understanding of the target. So when there is a very good understanding, those particular targets are known as the established targets. Now, these established targets, this does not imply that the mechanism of action of drugs that are thought to act through a particular established target is fully understood, but you have a fairly good understanding, good scientific understanding of that particular target. Rather, the established that relates directly to the amount of background information available on the target, in particular the functional information. Now, the more such information is available, the less investment is generally required to develop a therapeutic directed against that particular target. Now, the process of gathering such information, like such scientific understanding, having the lengthy publication history, this kind of the information, this process is actually known as the target validation in the pharmaceutical industry. Now, when we talk about the new targets, in general, the new targets are all those targets that are not established targets. The scientific understanding that is low, the length of the publication that is low. So all of those targets which are not established targets, they are known as the new targets. Now, these typically include newly discovered proteins, or proteins whose function has now become clear as a result of the basic scientific research. Now, the majority of the targets currently selected for drug discovery efforts are proteins, and the most important proteins, one is known as the G protein coupled receptor, and the other one, they are known as the protein kinases. Both of them, they are proteins, but these proteins, they are very important targets for drug discovery in the current era. If I give you the examples of the third drug target, the two examples, one is known as the G protein coupled receptor, specialized one, and the second one that is known as the protein kinase. The drug targets, what they do is that a biomolecular target, as I've told you, is mostly a protein or a nucleic acid, and this protein or nucleic acid, they are the key molecules which are involved in particular metabolic or signaling pathways. and they are actually associated with a specific disease condition or pathology, or the infectivity or survival of a molecular pathogen, of a microbial pathogen. Now, when you talk about the drug targets, what these drug targets do if they are interacting with the receptor. So these drugs, they may be the agonist for this receptor. What I mean by agonist is that these agonists are going for the activation of this particular receptor, or the drugs that can be antagonist for the receptor. That means that these antagonist drug, they are going to inhibit the function of that particular receptor. There can be activators for enzymes. There can be the inhibitors for the enzyme. So there can be drugs that can actually go for the activation of the enzyme in question, or there can be drugs, they are inhibitors for the enzyme in questions. There can be ion channel opener drugs, or there can be ion channel blocker drugs. So a variety of the drugs, they are available that actually target the receptors, the enzymes, or the ion channel. So if you like the video, please subscribe to my channel, hit the like button, and share it with your friends. In the next video, in the part 2 of this video, we will be actually focusing on the mechanisms, how the drugs they are developed.